Welcome to a short introduction to the Lipid Network Explorer. Here we're going to show you how to use LINEX and how to interpret the results and give you a little bit of an overview on how to use the platform. In order to do this, we're going to use one of the example datasets from our publication, which is the data from Wang et al., which is just shotgun lipidomics on colorectal cancer or colorectal cancer samples. You can also download some example lipidomics data by clicking on this link in case you want to try it out with our provided data, but you can of course also upload your own data. In order to upload your data, you need to go to the upload page. This is essentially separated into two parts. On the left hand side you can upload any files, and on the right side you can select options. So the first file that you have to upload, this is the only requirement, is the lipid dataset. So the lipid dataset essentially looks like this. Sample names are in rows and lipid names are columns, and each cell is a lipid value. Now the sample names need to either be converted into the lipid links nomenclature or follow the lipid links nomenclature, or be in any format that is supported by the lipid links nomenclature. We'll come back to this in a second. The second file that you can upload is sample data, meta sample data, which essentially just shows which groups each sample belongs to. So this is also a very simple file where the sample names are just the row names and the column gives the group assignments. Also, the column names here, so the sampling group, are not of any matter because we only read the two columns. The other files that you can upload are with respect to how we internally treat or compute the different connections between lipids. This is going a little bit too deep for this introductory tutorial, but you can read up in our paper what both the lipid class settings as well as the fatty acid settings mean. You could also look it up on our tutorial page here. And if you have any further questions or need any guidance as to how to use it, feel free to contact us. On the right hand side, you have different computational options to choose. So the first one is with respect to whether your data is log transformed or not. Generally, the data that you upload has to be pre-processed because we do not do any internal data processing. So if you have already log transformed your data, you can click this button. If you did not log transform your data, but you wish to do so, please click this button. Please do not click this button, sorry. The second parameter is whether or not you wish to compute log ratios. So if your data is not log transformed, you can choose whether to show log fold changes or just fold changes. If your data is already log transformed, this parameter does not have any influence. The next option to choose is whether to convert the names into lipid links nomenclature. So if your lipid annotation already follows this nomenclature, then you do not need to click this button. Also, it will take some time if you do so, even though they are already converted. But if you need to convert them, you can click this button and they will be converted internally. Also, if there are any errors, so if there are, for example, lipids that are not in any compatible format, these lipids will be reported afterwards and you can manually check them. The next option that you can choose is the Lipid Resolution. So this means which should be the highest resolution to consider. The default here is molecular species, which essentially means if you have MS2 data and you have molecular species, but also some species, then molecular species will always be treated as molecular species and some species as some species. And it's possible to also find the connections between the different levels. However, if you also have molecular species but do only wish to show some species, then you can select some species here. Theoretically, you can also select as n-specific. However, we only recommend to use this if you have, if your annotations are very good. Usually, molecular species should be sufficient to find what you're looking for. Other options below are focused on different statistical metrics. So basically the first two are whether you want to compute correlations and correlation changes and partial correlations or partial correlation changes between lipids, and what the significance threshold for correlation should be. So if you leave the default value of 0.05, then each edge that has a correlation between its lipids above, with a p-value above 0.05, it will be considered non-significant. Anything below will be considered significant. Now I'm going to untick partial correlations here just because it takes some time to compute, but you can of course always choose it. The other two parameters are just fold changes and p-values, and you can of course also pick which statistical test you want to use to compute the p-values. However, it's only possible to use binary statistical tests. For fold changes, or generally for both fold changes and p-values, you can also choose a reference group. And if this reference group is set, then only the binary comparisons between this reference group and all other groups will be computed. So if you have 3 sample groups, then you will only get 2 comparisons if you set any reference group. And it's important that these reference groups are exactly typed as they are in your sample group file, because otherwise the algorithm will not be able to match them. Once you've uploaded your data and set all your options, you can click Upload and Compute, which will guide you to this upload progress page. This normally takes a few seconds, and then you should see this green button here. There might also be error messages or warnings down here if you have empty cells, for example, so missing values. But yeah, unless you have a red error message, the computation worked fine. In order to show the network now, you can click on View Results, which will guide you to the analysis page. So once the network has loaded, you have multiple different options to choose from in order to visualize your network in different ways. So the default visualization is lipid node colors denoting the lipid class, and the edge colors denoting the type of reaction that connects the two lipids. The other lipid-related options you have to choose from are desaturation, chain length, carbon index, and double bond index. There are 3 different graph theoretical node measures, like closest centrality, between centrality, and degree centrality that you can choose. Or you can choose sample group related options if you uploaded sample group data before, which are the negative log 10 of the false discovery rate and fold changes. So for demonstrating purposes, we choose the log 10 fold change here, negative log 10, the negative log 10 FDR, and node sizes are fold changes. And then we're also going to look at edge color options. So edge color options are reaction types, correlations and correlation changes if you chose them before, and partial correlations and partial correlation changes if you had enough samples with respect to your feature numbers to compute p-values for partial correlations. So in this case, we're just going to set it to correlations for quick demonstration. And now if you have correlation values, the group annotation here will show you for which group the correlations are currently shown. So these are for normal mucosa. You can also select tumor, and then you'll see the correlations in tumor samples. Generally, the light gray is a correlation of 0, so basically no correlation. Red means a very high correlation, blue means a very negative correlation, or very high negative correlation. And you should also note that for this plot, the correlation values that are below the significance threshold that you selected on the upload page are set to 0. Now, in order to view different correlations and how correlations change between your sample groups, you can select correlation changes, and then comparison will show you which sample group comparison you're currently seeing. Because we only have 2 sample groups in this data set, there's only one comparison to show. And then you, for example, see here that there are different regions that have unchanged significant, completely insignificant values, or significances that change between different sample groups. In order to find any lipid species that you want, you can use the two fields here. The upper one, find lipid species, lets you look for a single lipid by its full name, for example TG481, or you can also choose one by substring, which would, for example, you can select TG to show all TGs, and then you will see all TGs being colored in this brownish color in order to show the selection. Now, if you, for example, want to show different reaction types, so basically these reaction types between lipids are either class related or fatty acid related, you can choose to only show one out of the two classes. If you want to know more about how or what these mean, you can look it up in the paper, and then you can only show either of the connections. You can also choose a significance threshold to mark which lipids have a negative log 10 FDR below or above a certain threshold, and you can choose whether they should be highlighted or not. If you click Enable Physics, you will see that once you, for example, change direction types, the network will rearrange in adaptation to the different edges that are present. And if you want to have a scale, basically have the legend scale being the same between different comparisons, so say you had 3 comparisons and they had different ranges of fold changes, for example, then you could click Unify Scales, and the scale would be the same, such that the colors are basically on the same scale for each comparison. Also, if you want to, for example, download the network with the visualization options that you have or that you're currently viewing, you can do this and click Download View. This will, I can do this real quick. Okay, apparently it's unavailable right now, but generally it gives you a PDF to download, and then you can view the exact same network with your legends as a PDF file and include it in your publications, for example. And this also brings us to the next page, which is the download page. So you have a few different options to choose from. You can choose to download the network as a standalone HTML file. This means you get the exact same functionality as on the analysis page, but you can actually send around the file and share it with your collaborators or keep it for later analyses. You can also download the network as a GraphML file, which is a general storage format for graphs, and this will allow you to, for example, use other visualization softwares like Cytoscape. Then you can download the legends as GraphML files. You can also download the class connections. This is especially interesting if you uploaded different class settings before. And you can also choose to download the matrices or for the different statistical metrics that we computed internally. And you can also download both the lipid species that were not converted or not convertible by lipid links, and your data as lipid links converted, such that if you want to run LINEX again, you don't have to convert them to lipid links again. And you can also download the color scheme for the lipid classes, such that you can include this or use it yourself. And after you've selected all files that you want to download, you can simply click download, and you will either be presented with a single file or with a zip file. So for further references with respect to the individual steps, and also with respect to the options that you have, you can consult the tutorial, or you can also contact us. Now, we generally store each data for 100 minutes, and then it will be automatically deleted. So we do not keep your data on our servers, but delete it just for data privacy and purposes. So if you want to delete your data right after you've done your analyses and downloaded everything, you can click delete now, but make sure to download everything before, because obviously they will not be available afterwards. If you want to know more about us, you can also look up our contact information on the About page. Here you can find how to reach us with questions, with suggestions or issues that you're experiencing. There you can also again find the link to our paper and the citation, where to find the code for it, and also a little cookie disclaimer. Thank you for listening, and we hope you enjoy the Lipid Network Explorer and you find it useful for your data analysis. And if you have any further questions or suggestions, feel free to contact us. So as a small addition to show how download view normally works, this is how it normally works, and then you can open it with any document viewer that you like, and then you'll have the network as with the coloring scheme and everything that you have on the web page, including all the legends that you can include in high resolution for your publications.